As we will learn also, proteins that are synthesized by the ribosomes don't sit around forever. Some proteins have long lives. Some proteins have lifetimes of 15 minutes before they're degraded, before they're turned over. One other distinction between our cells, that is the cells of metazoa and metaphyta, are the cell walls, analogous to the cell walls of bacteria, this green thing on the outside. As I said before, we do not have cell walls around our bacteria. And we will, as the semester goes on, go into more and more details about different aspects of this cytoarchitecture during the first half of the semester. Here, for example, is an artist's depiction of the endoplasmic reticulum. Why it has such a complex name, I can't tell you, but it does. It's called the ER in the patois of the street, the ER. And the endoplasmic reticulum is a series of membranes. Keep in mind that not the only membrane in the cell is the plasma membrane. Within the cytoplasm, there are literally hundreds of membranes which are folded up in different ways. Here you see them depicted. And one set of these membranes, often they're organized much like tubes, represents the membranes of the endoplasmic reticulum, which either lacks ribosomes attached to it or has these ribosomes attached to it, which causes this to be called the rough endoplasmic reticulum, to refer to its rough structure, which is created by the studding of ribosomes on the surface. As we will learn, just trying to give you a feeling for the geography of what we're going to talk about this semester, these ribosomes on the surface of the endoplasmic reticulum are dedicated to the task of making highly specialized proteins, which are either going to be dispatched to the surface of the cell, where they will be displayed on the cell surface, or actually secreted into the extracellular space. Many of the proteins that are destined for our body are not kept within cells, but are released into the extracellular space where they serve important functions. And so we're going to focus very much on them. Here's actually what some of these things look like in the electron microscope to see whether we can either believe or fully discredit the imaginations of the artist. Here's the rough endoplasmic reticulum. I showed you in schematic form before, and you can see why it's called rough. All these black dots are ribosomes attached on the outside. Here's the Golgi apparatus. You see these vesicles indicated here. And a vesicle, just to use a new word, is simply a membranous bag. And keep in mind, by the way, that we're not going to spend the semester with these highly descriptive discussions. Our intent today is to get a lot of these descriptive discussions out of the way so that we can begin to talk in a common parlance about many of the parts, the molecular parts of cells and organisms. Here's the mitochondrion, which we saw depicted before. It looks similar to, but not identical to our artist's description of that. And keep in mind that the mitochondria in our cells, as I said before, are the descendants of parasitic bacteria. Here's the endoplasmic reticulum and the way it would look as it does in certain parts of the cell when it doesn't have all of these ribosomes studded on the surface. The endoplasmic reticulum here is involved in making membranes. The endoplasmic reticulum here is involved in the synthesis and export of proteins to the cell surface and for secretion, as I mentioned before. Much of what we're going to talk about over the next days is going to be focused on the nucleus of the cell, that is on the chromosomes on the cell, and on the material which is called chromatin, which carries the genetic material. So the term chromatin is used in biology to refer simply to the mixture of DNA and proteins, which together constitutes the chromosomes. So chromatin has it within it, DNA, it has protein, and it has a little bit of RNA in it. And we're going to focus mostly on the DNA and the chromatin, because if we can begin to understand the way the DNA works and functions, many other aspects will flow from that. I mentioned the cell surface, and I just want to impress on you the fact that the plasma membrane of the cell is much more complicated than was depicted in these drawings that I showed you just before. If we had a way of visualizing the plasma membrane of a cell, we would discover that it's formed from lipids. We see such lipids there, phospholipids, many of them. We'll talk about them shortly. At the outside of the cell, there are many proteins, you see them here, which thread their way through the plasma membrane, have an extracellular and intracellular part. And these transmembrane proteins, which reach from outside to inside, represent a very important way by which the cell senses its environment. This plasma membrane, as we'll return to, represents a very effective barrier to segregate what's inside the cell from what's outside of the cell, to increase concentrations of certain biochemical entities. But at the same time, it creates a barrier to communication. And one of the things that cells have had to solve over the last 700-800 million years is ways by which the exterior of the cell is able to sense certain signals and transmit that information to the interior of the cell. At the same time, cells have had to invent a number of different proteins, some of them indicated here, which are able to transport materials from the outside of the cell into the cell, or vice versa. So the existence of the plasma membrane represents a boon to the cell in the sense that it's able to segregate what's on the inside from what's on the outside, but it represents an impediment to communication, which had to be solved, as well as an impediment to transport, and many of these transmembrane proteins are dedicated to solving those particular problems. Here you see, once again in artist's depiction form, aspects of the cytoskeleton of the cell. And when we talk about the cytoskeleton, we talk about this network of proteins, which, as I said before, gives the cell rigidity. Keep in mind that the prefix cyto, or suffix cyte, refers always to cells, allows the cell to have shape. And here you can see this network as depicted in one way, but here it's depicted actually much more dramatically. And here you can begin to see the complexity of what exists inside the cell. Here are these proteins. These are polymers of proteins called vimentin, which are present in very many mesenchymal cells. Here are microtubules made from another kind of protein. Here are microfilaments, in this case made of the molecule actin. And if we looked at individual molecules of actin, they would be invisible. This is end-to-end polymerization of many actin molecules. And we're looking here under the microscope from one end of the cell to the other end of the cell. And you can see how these molecules, they create stiffness, and they also enable the cell to contract and to move. Some people might think that the interior of the cell is just water with some molecules floating around them. But if you actually look at what's present in the cell, more than 50% of the volume is taken up by proteins. It's not simply an aqueous solvent where everything moves around freely. It's a very viscous slush, a mush, and it's quite difficult, therefore, for many cells to move around from one part of the cell to the other. Here you begin to get a feeling now for how the connection, which we'll reinforce shortly in great detail, between individual molecules and the cytoskeleton. And here you see these actin fibers. I showed them to you just moments ago, stretching from one cell, end of the cell to the other. And each of these little globules is a single actin monomer, which polymerize end to end and then form multi-strand aggregates to create the actin cytoskeleton. Here's an intermediate filament, and here's the microtubules that are formed. Once again, giving us this impression that the cell is actually highly organized, and that high degree of organization is able to give it some physical structure and shape and form. I think we're going to end today 2 minutes early. You probably won't object.